Hey children, welcome to Score 360 out of 360 in NEET Bio 2025 within just 250 days. So yes, today we are going to talk about fertilization and implantation, and I'll be talking about the concept for sure. Apart from that, the 5 PYQs and also certain extra questions. Why ma'am? Because I thought that while reading the NCERT and having all the check, I realized that there are certain questions which have come out of the box and you need an explanation for that as well. We never know what would come. So I thought, why not just include it over here so that it will be a great help for you. So what are we doing? We are doing human reproduction. And what did we do previously in the previous session? We spoke about the menstrual cycle, and there are certain things that I want to include even now in the menstrual cycle part. Okay, just few things. It's not including something else, it's just emphasizing on certain important facts, just for your knowledge. Yeah. And over here, this is how I taught in the previous session. So if you want a PDF of this particular class, you can join our WhatsApp channel and you can get the PDF there. Yeah. So let's talk about the menstrual cycle first, and then we will go on to fertilization and implantation. This particular class could be long, I'm not very sure. Okay, I'll try to pace it up as fast as I can, but in a way that you understand. But if it is fast, please bear with me, because I might be trying to explain you something very much important that you should not be probably losing out. Yeah. So while we spoke about menstrual cycle, there was one thing, that is maturation of follicles. Whenever the follicles, they were maturing, I told you when they come to this particular stage, they are releasing more amount of what? Estrogen or estrogen. Yeah. So the level of estrogen is high, and now when the level of estrogen is high, what happens? It goes and affects the FSH and LH. That's what we spoke about in that particular class. Now, a little bit of detailing over here is needed, because I thought that while solving other questions, you might get confused. So basically, when estrogen level is high, okay, it goes and tells the GnRH, that is gonadotropin releasing hormone, which is released by hypothalamus. It gives a signal to GnRH, gives a positive feedback that please release more of it. Okay, this happens, and which is why the amount of LH, okay, amount of LH also increases. Why children? Because basically, you know, there is hypothalamus. Hypothalamus releases GnRH. This GnRH goes and acts on the anterior pituitary. The anterior pituitary release LH and FSH, right? So as of now, I am saying the estrogen says that, oh, release GnRH, more amount of GnRH is fine. Now the GnRH will obviously go and act on anterior pituitary and make the release of this and this possible. So obviously LH has to increase, because if LH is not increasing, then ovulation would not be taking place. But you might have seen the FSH is not increasing that much. Why is that so? That is something that I wanted to tell. Okay, so FSH does not rise comparatively. Why? Because you remember the graphene follicles, basically the granulosa cells, they were also releasing something that is known as what? Inhibin. Now inhibin is going to act on FSH, which is why the amount of FSH will be comparatively lower than LH. I'm not saying entirely lower, comparatively lower than LH. Yeah, that was one thing. Now next thing, progesterone. Again, when I say progesterone, it is giving a negative feedback to GnRH. Again, it's GnRH, not directly on LH and FSH. Okay. Now when it goes and says, hey GnRH, stop producing your things. So when GnRH is not being produced in that much amount, so obviously it won't be giving anterior pituitary the signal. And now if anterior pituitary is not receiving the signal, obviously these guys would be made that much, which is why we see a fall in FSH and LH. There was corpus luteum. Corpus luteum was releasing progesterone. Progesterone is now going and saying GnRH, please stop. It gives a negative feedback. Please stop. Okay. Now, during this point of time, obviously LH and FSH will drop. If LH and FSH will drop, obviously the corpus luteum which was there, the LH was maintaining the corpus luteum. Now it will slowly start disintegrating or basically breaking down. Yeah. Now one more thing. Now corpus luteum, basically when we say that it is disintegrating, it is transforming into something that is known as corpus albicans. That's all. That's all I wanted to tell. So these are the extra points that you want. If you want, you can take a screenshot, or obviously you get the PDF as well. Yeah. Now we go ahead. What do we have? This was the question that was left to solve. Were you able to solve it, my little champions? If mammalian ovum fails to get fertilized, which one of the following is unlikely? Now over here I think the concept won't be tough, but the language that they have framed could be difficult for you to understand. So whenever you get these type of questions, try to cut the question down and then try to understand. So if mammalian ovum fails to get fertilized, so if fertilization is not happening, that is the egg and the sperm is not fusing, okay, which one of the following is unlikely? Which one of the following won't be taking place? That is the thing, that is the easiest question that they are asking. So if it is not getting fertilized, obviously corpus luteum, is there a need of it? No. So corpus luteum says tata bye bye and it goes. Okay. Now next thing, if corpus luteum is not being produced enough, the corpus luteum is disintegrating, then progesterone won't be produced enough. And same goes with estrogen. Estrogen will also be not produced enough. Yeah. But at the same time, as the level of everything goes down, we have studied that the follicles start regenerating because they again start to want to start the cycle. So the primary follicles that you see over here, they will obviously start regenerating. Okay, so corpus luteum will go down, it will disintegrate. Obviously if it is not there, progesterone won't be formed and as well as estrogen won't be formed. And at this point of time when everything is going down, obviously primary follicles start forming again. Okay, they grow again. Now over here, which is something that does not match? Let's see. So it says corpus luteum will disintegrate. Obviously it will disintegrate. So it is correct. Okay. Progesterone secretion rapidly declines. Obviously if it is not there, this also won't be there. This is also correct. Estrogen secretion further increases. Now how can that be possible? No, it's wrong. Estrogen is decreasing, right? It is decreasing. Now primary follicle starts developing. Obviously primary follicle is what? Developing. So what is the correct answer for this? This is the most unlikely event to occur. Okay. So I hope you were able to find the answer for this. This came in 2005. And now comes our dear fertilization. So let's read the NCERT first and then go ahead. I have a picture as well. I'll just keep it here. Yeah. So during copulation, what do you mean by copulation? Coitus or the sexual intercourse. Semen is released by the penis into the vagina. This process is known as insemination, obviously. Okay, insemination means the release of sperms into the female reproductive tract. Now, the motile sperm swim rapidly, passes through the cervix, enter into the uterus and finally reach the ampullary region. So, it is going fast. Let's see. So, over here it's saying that insemination happens, sperm will be released, it goes through the cervix and then where? Then through uterus and then it goes to the ampullary region. So this you remember the fallopian tube had three different regions, that is infundibulum, ampulla and isthmus. So it goes and waits in the ampulla. Okay, it goes to the ampulla because who is there in the ampullary region? The egg is there in the ampullary region, which is why the sperm is also going, or the sperm is not a full to go there. Okay, so over here the ovum released by the ovary is also transported to the ampullary region where fertilization takes place. So as of now you got a great piece of information which is something that you should never forget. Where does the fertilization take place? It takes place in the ampullary region. Children, please write it down somewhere. Okay, where does the fertilization take place? Fertilization takes place in the ampullary region. Fine. So when I used to study, it was ampullary isthmic region. So now they have made changes in the NCERT. It's just ampullary. So please go with ampullary. Okay. That is very much important. Now, the ovum released by that also we run. Now, fertilization can only occur if the ovum and the sperms are transported simultaneously to the ampullary region. So it's not like the egg comes and waits there and the sperm does not come at all. Then obviously, or comes after like a few days, then obviously fertilization won't be taking place. Because why? The egg will be just waiting for one day. It will be fertile for just one day. So during this 24 hours, if the sperm is coming and fertilizing, it's well and good. Or there can be chances that the sperm is already there. Sperm can remain inside the female's reproductive tract for almost 5 days. Yeah, but it remains viable for almost like 48 to 72 hours. Okay, make it 3 days. That's all. Okay, so basically at this point of time the sperm is already there and it has the viability period, and then comes the egg, then also fertilization can take place. But it has to come together. Okay, it's not like one will come this week and the other one will come other week, nothing will happen. So it's a very important thing. Now this statement might be you might be thinking, what kind of statement is this? Very simple to think of, but during exam, when it comes directly, we tend to forget things. So please remember this. Okay, this is the reason why not all copulations lead to fertilization and pregnancy. This is why not all sexual intercourse lead to what? Pregnancy. Because at this point of time, probably the woman might not be ovulating. Okay, or probably the sperm might have not reached this, the exact spot, the place as well. So either way, okay. So I hope this thing is clear to you. The only major thing that you need to remember is the site of fertilization is ampullary region, yeah, and also the sperm and the egg has to reach there simultaneously. Then only fertilization can take place. Now comes the next particular paragraph. We'll read it once and see what all we understand, and then obviously we have to go ahead. So the process of fusion of sperm with an ovum is known as fertilization. What is it? Sperm, ovum, fusion is known as fertilization. During fertilization, a sperm comes in contact with the zona pellucida layer of the ovum. So that means ovum has a layer, and what do you call this layer as? You call this layer as zona pellucida. So over here also they have made up a layer. That is this thing that you see in yellow as of now, yeah, this particular layer that you see, it is what? The zona pellucida layer, and induces changes in the membrane that block the entry of additional sperm. They are saying that the sperm comes in, fuses, okay, with what? Comes in contact with the zona pellucida layer, and then later does not allow any other sperm to come and fuse the egg. Okay, there are certain changes that are being made in the membrane. So we will see that everything, okay. Thus, it ensures that only one sperm can fertilize an ovum. So, at a time, obviously, a sperm can fertilize an ovum. So, other sperms, basically, they are set to say tata bye-bye and go. How and why, we will see. The secretions of the acrosome help the sperm enter into the cytoplasm of the ovum through the zona pellucida. So, we know that if you have seen the picture of the sperm that we already made, okay, at the top region, there is something that is known as acrosome. Now acrosome has enzymes which you call as what? Sperm lysins, right? Those that can break proteins and all of that. So basically it's saying that, the secretions of the acrosome help the sperm enter into the cytoplasm of the ovum. So to enter into the cytoplasm of the ovum, it's not very easy because there are so many layers. There is zona pellucida, there is outer layer also. We'll study about that. So you have to have some secretions over there, certain enzymes over there, only with the help of which you can get inside. So the secretions of the acrosome help the sperm enter into the cytoplasm of the ovum through the zona pellucida and the plasma membrane. So egg cell, it's a cell, so a cell will obviously be having a plasma membrane as well. Now this induces the completion of meiotic division of the secondary oocyte. So what information did you get over here? And this is the most important thing that I wanted to tell from here. This induces the completion of meiotic division of the secondary oocyte. That means the entry of sperm, the fusion of sperm and the egg, basically that induces the completion. Now the meiotic division is complete. So until now what we called as ovum was actually not ovum, it was secondary oocyte, right? What was it, children? If you remember, it was secondary oocyte which was stuck at metaphase 2. It had to complete its division. When was it going to complete its division? At fertilization. So during fertilization this division is completed, and now oocyte can give two cells. One will be bigger cell, one will be the smaller cell. This bigger cell you call as what, children? Ovum. This small cell you call as what? Second polar body. So that is what is exactly written over here. The second meiotic division is also unequal. It is not equal. One is big, one is small, okay? And results in the formation of second polar body and a haploid ovum. What do you mean by haploid? Haploid as in half the number of chromosomes are there. So this ovum will be having, if normal human cell has 46 number of chromosomes, so what is the half of 46? It is 23, because this particular cells that you see over here, they have underwent meiosis, and meiosis at the end of it, obviously you see the number of chromosome will be half. So ovum will be having just 23 number of chromosome, because sperm will also be having 23 number of chromosome. Together when they fuse, it's already 46 and everything is jingalala. Yes. Now The haploid nucleus of the sperm and that of the ovum fuse together to form a diploid zygote. How many chromosomes will there be in a zygote? You tell me. 23 plus 23 is what, children? It's 46. So the answer to the question is also there. Now, why did I stress, rather than just directly getting into the topic, why did I make you read this? Because I wanted to tell you something. You can get questions like this, okay, straight from your NCERT, even if you just read it out, okay. There is not much of conceptualization over here. It's a very simple question. In human females, meiosis 2 is not completed until what? So you know it's until what? Fertilization. No, it came in 2015. So it's as simple as that. So that's why I showed you, I wanted you to read everything. Now we will see the story of the sperm and the egg and the fusion, whatever I spoke until now. Zona pellucida, this and that and everything. So first we'll talk about sperm. So during insemination, that is when the penis enters the female genital tract, releases the sperms. How many number of sperms are being released during one ejaculation? It is around 200 to 300 million sperms. Okay, that many sperms are being released. But do all of them reach over there where the egg is waiting over here in the ampulla region? No. What happens? Let's see. So basically, sperms, they die, and there are 3 different reasons for the sperm deaths. Okay. First, the area, that is cervical region or vaginal region, everything comes out to be acidic in nature. Okay, so this particular area that you see is acidic in nature. Why? Why female genital tract have acidic nature? Because there could be entry of pathogens as well. There could be entry of foreign invaders which could harm the female body. Right? So obviously no one wants that, no one wants any sort of infection, which is why acidic pH is necessary, because at this pH most of the bacteria, organisms do not survive. Yeah, so that is one thing. So which is why poor sperm also die. Now cervical mucus is the next thing. So again, to plug into to stop the entry of any pathogen, again we have what? Cervical mucus, which could trap the sperms, and then the sperms cannot move, they won't be able to motile, they won't be able to move up. So this is one thing. Third thing, immune response. As I said, same thing, that was acid, it was providing acidic nature to prevent the entry of any pathogen. Similar to it, immune response, there will be so many cells which could kill the sperm, thinking that it's a foreign particle that is coming outside the body, obviously. So these are the 3 major reasons, which is why most of the sperms, they die. By most of the sperms, I mean that if you have 200 to 300 million sperms, only 200 of them could go ahead. So you see the number from million, that is 200 million, you just have 200 of them moving inside. Okay, now they have reached this region. Now, there is another very important thing that is going to happen in the female genital tract. So, I am saying in the female genital tract, I am not mentioning exactly in the uterus or in the fallopian, nothing. Together, I am saying is just female genital tract. The secretions of the female genital tract causes capacitation. So, you see, you are able to understand one thing, that is, there is some secretion happening inside the female genital tract which is acting on what? The sperm. And this particular thing causes was what? Capacitation. Now, what is the meaning of capacitation? Capacitation means you're making the sperm able to fertilize the ovum. You're making certain changes in the sperm that could help the sperm to get near the ovum and fertilize it. What kind of changes could that be? So, for example, you know that the whole sperm body, okay, that the whole sperm body is covered by what? Is covered by, any guesses, guys? Remember, we studied the plasma membrane. So you only think, if the plasma membrane is made weak by removal of certain proteins or certain making certain biochemical changes, now when this particular sperm reaches the egg, it will be very easy for the acrosome, which is at the top, to release its enzymes. And now when it releases its enzyme, it can obviously penetrate through the ovum and help fertilization occur. Now, if there is a very tough plasma membrane over there, can acrosome do its reaction properly? Obviously no. So this is one particular reason, which is why we need capacitation. okay, of the sperm. And who makes it occur? Basically the secretions of the female genital tract causes the capacitation. Okay, now capacitation, as I said, it includes removal of certain proteins and things from plasma membrane of the sperm. Apart from that, there could be other changes as well. Now once capacitation is done, it becomes easy for the sperm to fertilize. Easy for sperm to fertilize, and apart from that, it also increases the motility of the tail. Now the sperm will be able to move really fast. Okay, motility increases. So please do not judge me with the handwriting that I am having over here. I am not able to write like this. Okay, so basically, yeah, two major things for capacitation. Now while capacitation is happening, the sodium ions, there will be so many channels that will be opening up, and there will be intake of sodium ions inside. What happens? Intake of sodium ions happens, and this is very important. Why? You'll get to know really fast. Okay, so sodium ions are inside, just remember that. Now there is a story of the egg. That's all about the sperm. We need to talk about the egg as well. So over here, this particular structure that you see from here to here, we studied in the previous session that there is a secondary oocyte, okay, which is stuck in metaphase 2, and outside the secondary oocyte there will be zona pellucida, that is acellular layer. It is not made up of any cell, it is just made up of glycoproteins, lot of proteins, that's all. Okay, now outside that, what do we have? We have a layer of corona radiata. These are also cells, elongated granulosa cells only makes this particular thing. Okay, so basically what all do we have, children? We have the secondary oocyte, agree, SO I am writing. Okay, outside that, what do we have, children? We have this, why this collaborative D? Okay, outside this, what do we have? We have the zona pellucida, agree. Outside the zona pellucida, what do we have, children? Again, there should be something very much important, right? This particular thing that you see over here, the elongated ones, are you able to see? It is known as what? Corona radiata. Now children, one more thing, there is a space over here. Okay, there is a space over here. What do you call this? Perivitelline space. Perivitelline space. Okay, and obviously the ovum is an egg. Okay, secondary oocyte is an egg. So this particular layer that you see over here, it will be known as what? The plasma membrane. Okay, at least this much you need to, I am not telling you to remember Wittlein membrane, a lot of things. Okay, at least this much you can remember, right? So secondary oocyte is there, obviously it's a cell, it will have its own plasma membrane, and then outer to that you will be having certain other things, and also a perivitelline space, the space in between them, yeah, and then zona pellucida, and then what? The corona radiata. So when the graphene follicle, with the help of LH surge, when it happens, it bursts open and it releases this guy outside. Okay, I hope you are able to get it. So the secondary oocyte will be covered by all these things. Now let's go ahead. So this is how it looks. I don't need to label it again, I think. So this is what? Corona radiata. This is what? Zona pellucida, and this is our dear secondary oocyte, and these are our Chundamundu sperms. Yeah. So what happens to them? What happens to them? Okay. So listen to me very carefully now. The zona pellucida layer, okay, it has certain receptors which you call as ZP3 receptors. Zona pellucida 3 receptors. Now, the sperm will also be having certain proteins, okay, which could go and bind to these receptors. Now it realizes, oh, you are our team member. Now that means the fusion activity can be initiated. But there are so many sperms, they will be attaching on the ZP3 receptors, yeah, and the same reaction could take place. But obviously one or two may be faster, right? So basically what happens, once the ZP3 receptors and the proteins of the sperm, they bind together, there will be a set of reaction that is going to happen, which is known as acrosome reaction. What do you call this? Acrosome reaction, where basically what happens, children, The acrosome opens up and all the enzymes get released. What did the acrosome have until now? The sperm license. What did it have? The sperm license, right? Remember? Sperm license. Okay, you want an example? Remember the name of acrosin. What is it? Acrosin. What does it do? Acrosin basically, it cleaves the zona pellucida and trying to get inside. Okay, acrosin lyses the zona pellucida membrane and try to get inside, because here we have what? The secondary oocyte. So obviously this reaction will be taking place. It will be releasing enzymes over here and there, and then what will happen? This particular guy, that is acrosin, will be released. There will be another enzyme, that is hyaluronidase. Okay, that will also be released, which basically helps in what? Destructing anything that has the hyaluronic acid in them. For example, the granulosa cells, they have the matrix made up of hyaluronic acid. The zona pellucida also has a bit amount of hyaluronic acid. So everything of this sort will be cleaved. Okay, I'll write that also, hyaluronicidase. I might be wrong with the spelling, but yes, you can remember acrosin as of now for sure. Yeah, now that is done. Now one very important thing. Okay, how was acrosome able to do this thing? Okay, I agree, the sperm had a protein and the zona pellucida had a receptor and everything, but here comes the role of the sodium ions that we spoke about earlier. So I told you, right, that the sperm basically, while it undergoes the capacitation, what happens? One more thing happens, that is what? The entry of sodium ions. This sodium ions basically, once this particular point is reached, that is near the ovum or the basically secondary oocyte, at this point of time, it will also lead to influx, basically the entry of calcium ions. Okay, it will also lead to entry of calcium ions. Now this calcium ion is the one that initiate the process of what? Acrosome reaction, or basically the breakdown of acrosome. Okay, so that might not be very much important, but I saw an MCQ, I'll be explaining you that as well. So as of now, just remember it somewhere, that at least if you're not remembering the whole concept, the sodium ion is coming inside during capacitation, which is why the calcium ion also comes inside, and this calcium ion is something that could lead to acrosome reaction. Acrosome reaction means acrosome is bursting open, giving out all its hydrolytic enzymes over there, such as acrosin or hyaluronidase. My god, yes, and that. Okay, so yes, what does it do? It cleaves the zona pellucida. Now the zona pellucida is cleaved. Okay, what will happen? The sperm will go and try to fuse with the the secondary oocyte over there. Membranes of the sperm and the egg fuse and gets depolarized. What do you mean by that? So basically, I'll tell you. Membranes of the egg and the sperm fuse and gets depolarized. You need to cut all of these things down. Listen to me very, 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 very carefully. Okay. So over here, the first thing that happens is the sperm goes and attaches to the plasma membrane of the secondary oocyte. Now at this point of time, what will happen? There will be certain proteins that gets activated. Okay, there will be certain proteins that will get activated. Now during this, because of this activation, there will be certain ion channels that will now get open. What am I saying? Listen to me carefully. Sperm goes and attaches to the secondary oocyte on certain proteins. When it binds to certain proteins, what will happen? Just like how we turn on the switch, there will be so many ion channels that will be getting opened. Now what happens because of this ion channels? There will be entry of sodium ions. There will be entry of so many sodium ions. Agree? The sodium ions is also there. Now that happened. Okay. So there is plus. There is too much amount of sodium ions. Okay. Now what will happen? So there will be so much amount of sodium ions that will be getting inside. Now what happens? What is the problem with the sodium ions, or what could actually happen? For that, I need to rub a few things. Where can I get the thing? Over here. Yes. So see this very carefully. Okay. The sodium ion channels are opening up. There will be intake of sodium. Now, if there is an intake of sodium, there will be obviously positive ions, positive ions, positive ions inside, and this will bring out changes in the membrane of the oocyte. This particular entry of sodium ions will bring out changes in the membrane of the oocyte in a way that it won't allow any sperm to now come and fuse with the secondary oocyte. So this particular thing which occurs very fastly, you call this procedure as what? Fast block of polyspermy. You do not want so many sperms to come and fertilize the egg, right? Only one sperm and one egg. So basically, to prevent all the sperms that are going to come on the way, so what do you need to do? You need to bring out certain changes. What is that change? The sperm goes and stays near the fuses, binds to certain proteins that are present on the membrane of the secondary oocyte. Now when this happens, there will be so many ion channels that will be activated, and it will lead to intake of sodium ions inside. Now once it happens, once this depolarization happens, now it is more of positive, right? So it is depolarized, right? So at this point of time, there will be certain changes in the membrane, which is why it won't allow any other sperm to come and bind with the proteins over there. There will be certain biochemical changes. Now, can you stop the polyspermy because of just this? No, you need to have something more. So what can you do as an extra thing? So the next thing that happens is the sperm basically gives out the nucleus. Okay, the sperm also contributes its nucleus. The sperm nucleus is bigger than the sperm, please don't mind. Okay, so it gives out the nucleus. Now one more thing happens. Obviously it's a cell. If this is a cell, it will be having all different kind of organelles. For example, smooth endoplasmic reticulum as well. And smooth endoplasmic reticulum, if you remember, it is a storehouse of what? Calcium. It is a storehouse of what? Calcium. So now calcium is being released. Okay, there will be so much amount of calcium being released. Now what happens if calcium is being released? The calcium, it quickly goes and acts on what? Certain cortical granules that are present over here. By this time, I know that you might be feeling, oh, ma'am, what are you teaching? But trust me, it's going to be easy within 5 minutes. Okay, so over here, it will act on the cortical granules. Okay, the cortical granules have lot of things that could again bring out changes in the membrane. Okay, it has enzymes that can go and lyse something that is known as the ZP3 proteins as well. So basically these cortical granules are active. They go and slowly fuse with the membrane. Now everything which was there inside the cortical granules, which will be released outside in the perivitellin space, and further it will go and act on the ZP3 receptors which were present, which were allowing more and more sperm to come and attach and get entry into the whole structure, right? So if ZP3 itself is removed from that particular point, can more sperms enter? No, more sperms won't be able to enter. So over here what happens? The cortical granules releases its stuff. Yeah, it goes to the perivitellin space and then acts on the receptors or the proteins which are present on the zona pellucida layer and makes this whole layer very hard. Such that no sperm can fuse with it. And now this happens really slowly, which is why this method is known as what? Slow block of polyspermy. Okay, and now obviously I think that you need a revision, so let's just revise quickly, okay, without the help of diagram. Okay, so over here the first thing that happens is basically sperm binds with proteins on the membrane. Okay, on the membrane. Agree? Now because of this thing, what will happen? There will be sodium ion channels, sodium, that is Na plus, ion channels open up. Okay, now when sodium ion channels open up, this will cause this will cause depolarization. Now it will be too much amount of polarization, positive ions inside. Okay, now this change obviously will slowly make the sperm releases its nucleus into what? Into the cytoplasm of the egg. Into the cytoplasm of secondary oocyte. Now slowly what happens? The next step happens. That is what? These changes, okay, makes the smooth endoplasmic reticulum permeable, and it gives out what? Calcium ions. This calcium ions, what will it do? Again, in calcium ions act on what? Act on what are there inside the ovum? Cortical granules. Okay. Now please take a screenshot and then we'll go ahead. Please take a screenshot because I don't think that it will be coming in the PDF. Okay. So this is the thing. I hope this is clear. Okay. Now what will happen to the cortical granules? Cortical granules will be going and fusing on the membrane such that it will look like this. So if this is the membrane, the cortical granules go and fuse and releases all its content outside. Now once it releases all its contents outside, what will happen in the perivitral space? It will now go and affect the next layer, which is what? The zona pellucida layer, which I am going to make in green now. Okay, so this is the zona pellucida layer and this is the ZP, okay, it is the ZP3 protein. So basically this particular cortical granule secretion, it goes there, it makes this particular area lyrus. So these proteins, everything will be gone. If these proteins are not there, can more sperm try to fuse? Can they do acrosome reaction? Obviously no. So this is known as slow block of polyspermy, and this is known as what? Fast block of polyspermy, because when the depolarization happens, now there won't be no more sperm can come and there will be certain changes in the membrane. This will cause changes in the membrane such that no sperm can come and fuse. Okay, and apart from removing the proteins, that is ZP3 proteins, it will also harden the zona pellucida. Okay, I hope this is clear to you. So that was all about it. Okay, now yeah, so even if you are not able to understand things, because I think it's complicated, membranes of the sperm and the egg fuse and get depolarized. That is, the egg gets depolarized. Just a second. Egg membrane is depolarized and the nucleus of the sperm also enters the egg cytoplasm. Now if the egg cytoplasm and the nucleus, everything is there, obviously it will lead to what? Fusion, right? So this is the cortical granules that I was trying to make. I already showed you. So cortical granules are also released in the perivitral space, which goes and act on the zona pellucida layer, as I already told you. Yeah, and the zona pellucida hardens. So if you see the structure, this is how the fusion occurs. It gives out, the sperm gives out the nucleus. Okay, it is known as male pronucleus. Okay, what do you call this? You call this nucleus as male pronuclei. Okay, now there will be obviously the secondary oocyte is also having nucleus and it is going to divide. Now when the division takes place, what will happen? There will be a set of chromosomes being separated there, there will be a set of chromosomes being separated over here, such that there will be two cells. One cell will be over here, small cell, and the other cell will be over here. It is the big cell, or what do you call as ovum, right? So this particular thing that you see, very small cell that you see over here, if I try to make it more in a more better manner, it looks like this. So this is the small cell and this will be the big cell that will be there. Over here, this is the nucleus of the what? The female nuclei, right? The ovum at last. So female pronuclei. And this is what? The second polar body. So you have the ovum over here, which will now, obviously, they both will fuse, and together when they fuse, what do they give? They give zygote, which is a single cell having 46 number of chromosome. Is zygote diploid or haploid? It is obviously diploid. It is not haploid because the sperm was haploid, yeah, and the egg was also haploid. Together when they fuse, they will be giving a diploid zygote with 46 number of chromosomes. I hope this is clear. Now here, when fertilization takes place at this point of time, when all this thing happen, that is the point where the meiotic division completely finishes up. Yeah, so that's all I wanted to tell you. And now comes the portion of questions. So over here, is there any doubt that you might be arising? Please write it down somewhere so that you can ask me in the comment section if you want to. Okay, and if everything is sorted, then it's all good. Okay, so I'll just have a quick revision, just to okay, because I am not self-satisfied with what happened. So let's just quickly have a small revision. Do not think of anything else. So first thing that we spoke about was the sperm. How many sperms were releasing? Almost 200 to 300 millions of sperms were releasing. But could all of them go inside the female genital tract? Were they able to surpass the whole reaction and go to the the secondary oocyte, which was there in the ampullary region? No. Why? Because of three reasons. First thing, acidic nature of the female genital tract. Second thing, cervical mucus, it could trap the sperms. Third thing, because of the immune response, there will be so many cells to kill the invaders over there. Now only 200 of them got inside. Now once they get in, what will happen to the sperm? They will undergo a process which is known as capacitation. What do you call this? Capacitation. What do you mean by that? You bring out certain changes in the sperm that will help the sperm to fertilize the egg. What kind of changes, ma'am? Basically changes in the structure of the plasma membrane. You can remove certain proteins, have some biochemical changes such that it won't be difficult for the acrosome to release out its enzyme later on when the sperm and the egg fuse. Okay, so that is what you mean by capacitation, and definitely that will take place. So during capacitation, there will be so many sodium ions that gets inside. This again causes depolarization in what? In the sperm as well. Yeah, and now the sodium ions will later cause what's a release of calcium ions also. Okay, it will take calcium ions also inside. Once calcium ions are inside, they are going to initiate acrosome reaction. So let's not talk about that as of now. Come over here. As of now, let's talk about the sperm that has went towards the egg. Now, once it goes to the egg, it sees that there is corona radiata, there is zona pellucida and lot of things. So, but sperms also have certain proteins which goes and fuses. Okay, which goes and binds with the proteins of the zona pellucida layer, which is known as ZP3 protein or ZP3 receptor. Now, once it binds, what happens? The acrosome reaction initiates. That is, the acrosome will slowly burst open and release out all the sperm lysins, the enzymes. One such enzyme was acrosin, which is going to cleave the zona pellucida and get inside the next space, that is perivitelline space, and then obviously to the secondary oocyte. Now, all the sperms are running towards fusion. Right, so obviously not all of them can go there. One particular sperm, the first one to reach there, okay, which has all the good properties, it goes and binds on the proteins which are present on the membrane of the egg. Now once binding has taken place, at this point of time it will bring out so many ion channels. It will open up so many ion channels which is there around the membrane. Now when the ion channels are opened, what will happen? The sodium ions gets inside. The sodium ions, once it gets inside, it becomes, the whole membrane becomes depolarized. There will be certain changes in the membrane such that there won't be so many sperms that could come and attach or try to fuse with the egg itself. Okay, but this is fast block. This could be temporary. There should be a permanent change as well. So what could be done? After this happens, the nucleus also gets inside. Okay, inside the ovum, and also the calcium ions, okay, the calcium ions which were there in the smooth endoplasmic reticulum, they also come outside. Now this calcium ions goes and tells the cortical granules to, hey, go these cortical granules have so many enzymes which could cleave what? The ZP3 receptors which are there on the zona pellucida layer. Hey, it says that, hey, please go fuse with the egg membrane, go outside, go into the perivitelline space and then act on the ZP3 receptors and bring out certain changes in the zona pellucida layer such that it becomes hard, so that no other sperm can get inside or try to fuse. Now once it happens, after everything is done, basically simultaneously what is happening, the nuclei, the sperm and the egg is fusing. But before that, the secondary oocyte is completing its meiotic division. Which one? Second meiotic division, right? And now once it divides, what do you get? A ovum and the second polar body. Now ovum has a nucleus, the sperm has a nucleus, both the pronuclei they fuse to become what? Zygote. I hope now it's clear. Okay. Now children, the first question. Capacitation occurs in. Where does capacitation occurs in? And I already told you, please do not remember areas like uterus or this and that. No, it's just the female genital tract or the female reproductive tract, nothing else. So capacitation does not occur in the epidermis, in the vas deferens or the rete testis. It is always the female reproductive tract. It came in NEET 2017. Going ahead, what do we have? Fertilizant is a chemical substance produced from. Now this is the question that I was talking about, certain extra things, right? So fertilizant is a chemical substance which is produced from the mature eggs. Why is it for? What is the use of it? So for the egg to recognize that the sperm is also of the same species. For example, human sperm and human egg are only fusing. You have certain proteins. One such protein is there, that is fertilizant. It's a chemical substance that is being released, and the sperm will also have anti-fertilizant. Both of them obviously will bind, fuse and things like that happens. So basically it helps in the recognition of whether it is species specific or whether the egg and the sperm are of the same species. If it is not of the same species, obviously this particular fusion would not be taking place. Okay, so it is released by the mature eggs. This is something extra. This is something that you should have to remember. It came in 1997 and also 1991, and it could be repeated anytime. And it's not just 1997 and 1991. Even in 2000 also it was repeated, and there could be a question coming in your test series as well. Okay, from fertilization. So be prepared. So it's a chemical substance which is produced from the mature egg, so that the species recognition could be done very well. Yeah. And also it helps to stay, it helps the sperm to come and bind to the egg properly. Okay, that is also one major use of fertilizing. Going ahead, we have a question. Acrosome reaction in sperm is triggered by. The reaction of acrosome in the sperm is triggered by what? Capacitation, release of lysine. Lysine is the enzyme that is released by the sperm itself, acrosome itself. Release of fertilizing, or what? A, capacitation. B, release of lysine. C, influx of sodium ions. And D, release of fertilizer. Chal. So this question I want you to research. Okay, this is kind of a homework for you. You can try to find it out. If you are able to do it, do let me know. If you're not able to do it, then also do let me know. I'll try to explain you in the next session, but not here. So this time I'll probably give you two questions. One this, and the other one will be, there will be one more question. Okay, so please try to do it once. You might have to do a bit amount of research, or if you understood acrosome reaction, then you might have got the answer as well. But still, I want you to write the answer and your explanation as well. Yeah. Now that is done. The fusion fertilization is done. Okay. Now let's go on to the next thing, that is the sex determination. So one has to remember that the sex of the baby has been decided at this stage itself, that is during zygote formation itself. Yeah. Let us see how. As you know, the chromosome pattern in human female is XX. Do you know that? We have studied in 10th standard, right? So females, they have two sex chromosomes. Okay. So sorry. Why does this happen? Yes. This is X and this will also be X. And the males will have, will also have a chromosome. It's kind of different. This is X and this is Y. The Y chromosome is smaller. Does this also look like X? Just a second. Okay. Now, Okay, so the human female is XX and the human male is XY. Cool. Therefore, all the haploid gametes ova produced by the female have the same sex chromosome, that is X. Whereas the male gametes, the sex chromosome would be either X or Y. What do you mean by that? When we say half the number of chromosome goes, that means either this one will go, that means either this one will go or this one will be going from the female's end. But on the male's end, either this could go, either this could go. So, just think, a female is making ova. Do they have any choice? Are they allowed to have X and Y chromosome? No. The genetics does not work like that. They only have X chromosome to contribute. They do not have anything. Okay. So, suppose the female gave this X chromosome or he gave this X chromosome. The same thing. The egg anyways will be having X chromosome. This is the egg of the female. But if we talk about males, okay, if sperm is being released, the sperm can have either X chromosome or the sperm can have either Y chromosome because they have a choice over there in their genetic makeup. It's not like a men also can definitely choose which chromosome he should send. But basically this is how the genetics is being built. Okay, so they also have a choice, that is X and Y. They only have a choice, that is X and Y. Now suppose the X sperm came and the egg obviously has X chromosome. Both of them fuses. What will happen? The child will be XX. So basically I'll just make it over here. So if the egg is having X, the sperm is also having X. The child, when it fuses, will be having XS. That means the child will be what? A girl child. Now on the other hand, the female obviously again it just has one option to give out X chromosome. But the sperm, if it has Y chromosome, when both of them they fuse, what do you get? X and Y. Now X and Y is the chromosomal structure for what? Men, boys. So this child will be a boy. Now it was said in the previous, like even now at some places where education has not been reached yet, at this point of time also they think that females are responsible for the sex of the child. I will say no one is responsible for the sex of the child because we are not the one who have designed our genetics. So it's not either men or nor women, that is us, who is responsible for the sex of the child. It just is the magic of the nature. It just happens, right? So yes, this is a very important question. Again in NEET it has come. You might think, how it's so simple, then why do they actually give? They also have to give certain easy questions, right? So yes. So where were we? Okay, therefore all the haploid gametes produced by the female have the sex chromosome X, whereas the male gamete have the sex chromosomes could be either X or either Y. Hence 50% of the sperms carry X chromosome and 50% of them will be carrying Y chromosome. After fusion of the male and the female gametes, the zygote would carry either XX or XY depending on whether the sperm was carrying X or Y. Okay, the zygote carrying XX would be developing into a female, XY would be developing into a male. You will learn more about the chromosomal everything in the next chapter, in chapter number 5. That is why scientifically it is correct to say that the sex of the baby is determined by the father and not by the mother. So it is scientifically, obviously scientifically we can say that the sex of the child is determined by the male partner. Why? Because they have X and Y chromosome. But is it good to blame anyone regardless of their sex? No, you should not be blaming. Okay, so my children who have attended this class, please make sure that you are not blaming anyone. And if you see someone who is blaming either of the genders for sex of the child, first thing, you should be okay with any sex, right? It should not be like male or female, whoever comes your way. Please just be happy for that rather than complaining, right? So if you see someone, just tell, hey, do not do this. This is how it works. You can try to explain. This is why education is being given to you. Yeah. So in our society, in NEET 2013 it came, okay. In our society, women are blamed for producing female children. Choose the correct answer for the sex determination in humans. What is the correct way of sex determination in humans? Due to some defect like aspermia in man. Okay. Aspermia as in there is no sperms being produced, okay. Due to genetic makeup of the particular sperm which fertilizes the egg, okay. Due to genetic makeup of the egg, but the egg just has X chromosome, so this is wrong. Due to some defects in the woman, again this is wrong. The sex would not be determined by the defects in the women. Due to some defect like spermia in man? No. What is it? Due to the genetic makeup of a particular sperm which fertilizes the egg. If the sperm has X, it's a girl child. If the sperm has Y, it's a boy child. That's all. As simple as that. So this is the correct answer. Okay. Going ahead, what do we have? We have implantation, which is the last topic for today. I hope after this we can all rest for some time. So let's read it first, children, and then we'll go ahead. The mitotic division starts as the zygote moves towards the isthmus of the oviduct called cleavage towards the uterus. That is, it is saying the zygote has now been formed. It is a single cell. Always remember, it is what? A single cell structure. Now over here, the zygote has to divide itself. It has to increase its number, the cells, right? So obviously, when the zygote, the cell, it divides, can it be meiotic division or meiosis? No. Why? Because we do not have to half the number of chromosome. The zygote has 46 number of chromosome. After division, the cells that it might give rise to should also have 46 number of chromosome. You have to give a whole organism, right? You cannot have the number of chromosome over there. Zygote is diploid, so other cells should also be diploid. Okay, so they undergo mitosis, and while they do this, the zygote which was there in the ampler region, because the fertilization took place there, right? So now the zygote slowly divides itself and moves towards the isthmus and then to the uterus. Okay, this process of division and going towards the uterus is known as cleavage. Basically, this process of division is known as cleavage. What do you call it? Cleavage. Okay, and forms 2, 4, 8, 16 daughter cells, as in the 2, 1 will divide into 4, 4 will divide into 8, 8 into 16. That's what they say. And all these cells that are being formed, what do you call one cell as? You call this one cell as blastomere. You call this one cell as blastomere. Okay. The embryo with 8 to 16 blastomere is called morula. Now, what is embryo, ma'am? So, basically, once the zygote, which was single celled, it starts dividing. Now, from this point onwards, it is known as an embryo. If it's 2 cell, it's an embryo. 3 cell, embryo. Whatever cell, it's just embryo, not 3 cell, 4 cell. Okay, so it will be always an embryo. Now, they are saying there is a stage in the embryonic phase. Okay. That is where the embryo will be having 8 to 16 blastomeres. Now, during this phase, the embryo has a separate name. What do you call it? You call it morula. How many number of cells do they have? How many number of blastomeres do they have? 8 to 16. 8 to 16, double the 8 is 16. Okay, so 8 to 16 number of blastomeres are there in morula. Morula is what? A phase, a stage of embryonic development. That's all. Okay, the morula continues to divide and transforms into blastocyst. Now, this is the next stage of embryonic development. After morula, after it divides a lot, later comes what? A stage which is known as blastocyst stage. So, morula is there, blastocyst is there. As it moves further into the uterus, the blastomeres in the blastocyst are arranged into an outer layer known as trophoblast. So, you can just imagine that if there are certain cells, they arrange themselves into outside, there is a layer, and what do you call this layer as? You call this layer as trophoblast. And what? Inner group of cells attached to the trophoblast, and there will be certain cells which will be a yolk. So, if this is the layer of cells, then there will be some layer of cells over here which are being attached to this particular layer. What do you call this particular layer as? This is trophoblast, and this mass of cells you call as inner cell mass that you see over here. Don't worry, I have a picture of it. Okay, the trophoblast layer then gets attached to the endometrium, and inner cell mass get differentiated as an embryo. Now, what happens? Basically, as I said, the division is happening, and who is waiting? Who is waiting for this particular embryo? It is the endometrium layer of the uterus. Remember the uterus having the blood layer, the thick blood layer, glandular layer being present over there. What is that? Endometrium. So, slowly this guy goes and attaches to the endometrium. That is what it is being said over here. Yeah. And the inner cell mass, this particular thing that you see over here inside, it will be giving rise to the baby, the fetus. Okay. The whole body of the fetus. Whereas the trophoblast, the outer layer, will be giving rise to what? Placenta. It will help in the formation of the placenta, which is now later going to provide nourishment to the growing embryo. Later stage, okay, not as of now. So, the trophoblast layer then gets attached to the endometrium, and the inner cell mass gets differentiated as embryo. After attachment, the uterine cells divide rapidly and covers the blastocyst. So basically the layer of the endometrium, it slowly covers the the whole thing that you see over here, blastocyst. Okay, and I know that you're not able to understand a lot of things, but I'll try to explain. Okay, as a result, the blastocyst becomes embedded in the endometrium. Now it's totally inside the endometrium, and this is known as implantation. This is known as implantation, and it leads to pregnancies. Now if it sustains, obviously it will lead to pregnancy. Are you ready to see it? Let's see it. So over here we have zygote, yeah, corona redita, just forget it. Okay, what do we have? We have the zona pellucida layer still intact. This red color thing that you see over here is the zona pellucida. Agree? Zygote is single celled. Agree? One nucleus. Okay. This zygote undergoes division, that is mitosis. Any other division? No. Just what do you call this division as? You call this division as mitosis, or you call it as cleavage. What do you call it as? Cleavage. Now there is a specific detail in cleavage, I'll tell you that. So when one zygote divides into two, there are two cells now. What do you call these cells as? You call these cells as blastomere. What do you call this cell as? You call them as blastomere. During this particular point of time, you need to remember, whenever cleavage happens, the cell size decreases. Please remember, it has come in NEET. Okay, the cell size, it was big, it has now decreased in the size when cleavage is happening. Okay, it happens within 30 hours, that is 2 cell stage it reaches. Now 4 cell stage it reaches within 60 hours, and then it will keep on dividing. Okay, then comes a stage which is known as morula, where almost the number of cells or the blastomeres is around 8 to 16. It will reach morula stage when on the 3rd or the 4th day, basically the 4th day. We say on the 4th day. By 4th day it will be around here. Okay, it will be like, it will be in the ampullary ischemic region. Slowly it will be moving down and staying in the uterine cavity. It reaches to the uterine cavity. Okay, now what happens? The endometrium layer, as you know, is already there. It will start releasing what? Glycogen, because it knows that this embryo is going to embed and it has to provide nourishment to the embryo, right? So obviously glycogen will be starting to get released. Remember, when we studied about menstrual cycle over there, we called the last phase as secretory phase. Why? Because at this point of time, what was happening? The endometrium started secreting the glycogen. So the glycogen is being used over here. I hope you got it. So on the 3rd and 4th day, morula is made, and slowly it comes down. Okay, divides. Almost 100 cell stage it reaches over here. It comes to the uterine cavity, and now what happens? Let's see. So it has come. The endometrium is secreting glycogen. Slowly, the glycogen is taken up by the embryo, the growing embryo that is being seen over here. And as you can see over here, there will be a cavity that will be formed, which will be having all the glycogen being stored. This particular cavity that you see, you call as blastocele. What do you call this as? You call this as blastocele. And as I told you, the cells, they have an outer layer. What do you call this outer layer as? Trophoblast. And then there will be inner cell mass. Okay, just known as inner cell mass. What will happen to the inner cell mass? Inner cell mass is something that is going to actually make the embryo, and the trophoblast is going to get embedded in the entire layer of endometrium and form the placenta, help in the formation of what? The placenta later on. Well, this stage that you see over here, where there is a blastocele in between, there is inner cell mass, there is trophoblast and everything. What do you call this stage as? You call this stage as blastocyst stage. So at blastocyst stage, the implantation takes place, and when does it happen? On the 7th day. On the 7th day, implantation happens. Okay, so we know on the 14th day ovulation takes place, right? And then the corpus luteum is there, which releases progesterone. The progesterone makes the endometrium line very thick. At this point of time, by the 7th day, the implantation has taken place. What do you mean by implantation? Basically, this particular structure that you see goes slowly inside the endometrium layer and gets implanted. Okay, just a second. And now what happens? It goes stuck there, and slowly this particular layer takes everything inside. Now the whole thing, the blastocyst, is inside the endometrium, and here the trophoblast, it will be releasing something that is known as SCG. What is SCG, children? Human chorionic gonadotrophin. That is something that is a sign for pregnancy. So people who use pregnancy kit, okay, they check for SCG level in the urine. SCG is one that makes the lines appear. So over there, SCG is a very important factor which starts to get released just the day after implantation, that is on the 8th day. On the 8th day, the trophoblast will be releasing HCG. And why actually women have HCG? It helps in maintaining the corpus luteum. So what happens, if you remember, during menstrual cycle, on the 14th day, the corpus luteum was there, right? The corpus luteum releases all things, that is the progesterone and everything. The progesterone, okay, it maintains the endometrial lining. When the progesterone goes and acts on the GnRH, the FSH and LH level goes down. Now when LH level goes down, obviously this guy, the corpus luteum, will slowly start dying off. Okay, by 7 days it will stay, and then it will slowly start dying off. Do we need that? No. This time pregnancy is happening. So this was the case where pregnancy was not happening. At this point of time, HCG will be released by the trophoblast, okay, during the 7 days. SCG, when it is getting released, this SCG will now go and act on corpus luteum such that it sustains there. It does not die. So it does not need LH now. It is okay with HCG over here. So SCG will be helping in maintaining corpus luteum. Now if corpus luteum is there, progesterone will be there. Yeah, and the progesterone can help in maintaining the endometrium layer over there intact. I hope you got this. So that's all. It's very, very, very simple. Yeah. So as of now, what do you need to remember? Please remember, morula is formed on 3rd or 4th day, basically 4th day. How many cells are there? 8 to 16 cells are there. Yeah. Going ahead, what do we have? You have blastocyst stage, okay. When an implantation happens in this particular stage only on the 7th day. On the 7th day, the implantation takes place. The outer layer is trophoblast, inner layer is inner cell mass. Done. And the trophoblast is responsible for releasing HCG, human chorionic gonadotrophin, which maintains the corpus luteum. Now, if corpus luteum is there, progesterone is there, that means endometrium lining will be there. So as of now, until the time the placenta is formed, it takes some time for the placenta to be formed, few months. So during this time, obviously there should be someone who could provide nourishment. Who is that? Someone that is endometrium layer. So to maintain that, we need all this thing. Yeah, and cleavage is a division through which, with the help of which, the cell size decreases. Okay, please remember that. And they are just undergoing mitosis, nothing else. Yeah, so I think we have covered inner cell mass, embryo. Remember that. Now, in human female, the blastocyst forms placenta even before implantation. Is it? No. Placenta is formed after some time. Okay, gets implanted into the uterus 3 days after ovulation. No. We saw that implantation takes place on the 7th day. So that is also wrong. Gets nutrition from the uterine endometrial secretion only after implantation. Is it so? It already received nutrition before implantation, right? Glycogen got inside in the blastocyst, in the blastocele already before implantation, right? So this is also wrong. Gets implanted in the endometrium by the trophoblast cells. So is it right? Yes. Trophoblast cells are the ones that is responsible for implantation. It goes and slowly gets implanted over there. Yeah. So this is the correct answer. Now, identify the human developmental stage shown as well as the related right place of the occurrence in the normal pregnant woman and select the right option for the two together. Okay. So, related right place of its occurrence in a normal pregnant woman and select the right option for the two. Let's see. Developmental stage and site of occurrence. Late morula. Blastula. Blastocyst. A cell morula. Tell. Where is it? So, easiest thing, you have to identify the Pakka one that has everything correct. Blastocyst in the uterine wall. Is it correct? Obviously, it has to be correct. Yeah. So that is the correct answer. We just studied it right now. So this picture that you see is of blastocyst, and it is there in the uterine wall. Done. So is there someone who got all the answers correctly? These were the 5 questions that I gave you. Capacitation occurs in where, children? In the female reproductive tract, nowhere else. Remember, right? It's the changes in the sperm that will help in the fertilization of the ovum. Acrosome reaction, this is for you again. Fertilization is a chemical substance produced from. Fertilization is a chemical substance produced from what? Mature eggs. And now, in our society, women are blamed for producing female children. Is it correct? What is responsible for it? It is due to genetic makeup of the particular sperm. It can have X or Y. If it has Y, then XY brings out male child, and XS, then it's female child. In human female, the blastocyst. In human female, the blastocyst forms what? Tell me. D. Gets implanted in the endometrium by the trophoblast cells. So since I gave you one question, I think I can give you one question. You can solve it right now. Okay. Sperm and the egg nuclei fuse due to. Why do the sperm and the egg nuclei fuse? Any guesses? This was supposed to be the homework, or let it be the homework. You can try for yourself. Base pairing of the DNA and RNA, formation of hydrogen bonds, mutual attraction due to difference in the electric charges, attraction of their protoplast. So I'll give you a clue. Protoplast as in there is no cell wall, there is just cell membrane and the cell. That is what you mean by protoplast. Okay, now you can think for the answer. That is the clue that I had to give you. And also please solve this particular question that you see over here. Acrosome reaction in the sperm is triggered by capacitation, release of lysine, influx of Na, and release of fertilization. Basically, or else I will tell, it's influx of sodium ions. That is the correct answer. Influx of sodium ions which later lead to what? Basically influx of calcium ions, and then it will lead to acrosome reaction in there. Okay, this is thing that we learned previously. So these are the 5 questions. And if there is anyone who have got all the 5 questions correctly, do not forget to comment down at least 4. Okay, and this question is for you, definitely. Do not forget to solve it. And the next session will be on gestation as well as parturition. Okay, gestation, and we will see a bit of parturition and stuff like that, and then we'll finish it up. So that's all. Do not forget to subscribe to the channel and have a really good day.